I'm Arnold Levine. I'm a professor at the Institute for Advanced Study in the Simon Center for Systems Biology. So I'm one of the biologists here at the Institute, and I've helped to organize this course. So if you have a good time, you can thank me, and if you have a terrible time, you can come and complain. I don't mind. Let me just say a few words about the course and a few words about who you are and a few words about who the faculty is. First of all, there are 65 students who are taking this course. It's quite a remarkable, diverse group. The great majority of the students are in graduate school or in postdocs in physics. Then there's another group of about 10 or 12 or 15, depending on the day, of people who are either biologists working in biology labs and actually doing research on topics that we're going to be talking about, or are physicists who have come into biology and are taking this course as well. So you're a very diverse group. And we've tried to respond to that, and I'll tell you a little bit about how we've organized the course to respond to it. It means that sometimes you may be bored and sometimes you may be challenged, and hopefully there's enough in the middle that makes you interested in what we're talking about. The diversity continues. That you're remarkable, come from 48 different institutions from around the world and 24 different countries. So what was great about this course, this is a course, by the way, that has been given for 20 years here at the Institute for Advanced Study. What's different this year is the topic. Usually it's physics for physicists. And this year it's really biology and physics and computer sciences and math for physicists as well. And it recognizes something that the Institute recognized back about the year 2000, and that is that biology has started to develop some really interesting questions and develop it in quantitative ways that physicists and mathematicians and computer scientists and engineers could in fact actually come into biology and make major contributions in biology. There are an awful lot of large data sets that you'll hear about that come from DNA sequencing and from a whole variety of other bits of information about life processes for which the analysis of the data sets is very challenging and very interesting and has errors in it and so forth and so on, and is really good food for thought for people in physics. And it was that recognition that made the physicists this year come to us in the Simon Center Systems Biology and say, hey, why don't we put on a course and see how it goes for physicists, mainly for physicists, that would explain what are interesting questions in biology. Right? So the first thing to say about this course is that the topics are not systematically biology or anything like that. They are really interesting questions in biology. And today is really an introduction to that topic. One of the things we do rather routinely, and you'll see in almost every talk subsequently after today, is rely on DNA sequencing a lot. And so today we're going to talk about DNA structure, DNA sequencing, genome structure. We're going to talk about, we're going to even address the question about what are the minimal number of units or genes that in fact can make up a free living organism. What does genome size mean? What do genomes really look like? And so today's a genome day, right? And it's really meant to be a bit introductory, but we're going to try to push it like we do for everything else. A typical day may be a topic. For instance, we'll follow that by evolution. That's really a very interesting topic for mathematical analysis, in fact. And we'll follow that by viruses, which are the smallest of chromosomes and the simplest of organisms. They're obligate parasites, so they only grow in cells, and they rely upon the cell for their replication. We'll follow that actually by a discussion on cancer, that is cell growth and the regulation of cell growth. And we'll follow that with immunology, which is a really interesting topic about the memory and specificity and a whole bunch of questions that biology addresses that overlaps with physics. And we'll just continue with that. I mean, episodically we'll have a lecture for example, on circadian rhythm, which is the mechanism by which all cells, and I really mean all living cells, bacteria all the way through human cells, every one of your cells, measures time, biological time. And there's a whole process to measure biological time, and it turns out to be a really important kind of question for biology related to a number of things in life processes. So as we sail through these topics, right, normally the first lecture will be given by a biologist, and then we'll slip into the second and third lecture, which could be given by people who are have come from the physics community and do biology. So of our faculty, there are 28 faculty that'll be coming over the next 2 weeks. And of that group, about 12 to 15 of them have been trained in physics, mathematics, computer sciences, or engineering. And so they have their doctorates in that, they have their postdocs usually in that, and then they come into biology and they do very quantitative work in biology. So a typical day will flow from the biology through the physics, if you will, of kinds of questions that we could ask, right? All right, and then we're going to end up that we've decided on the last 3 days that there are very interesting episodic kinds of people to have and topics to cover. So it isn't going to be so coordinated that the whole day will be a theme. The last 2 or 3 days will be individuals who are studying interesting topics. Among them will be epigenetics, which is really reaching its own peak and prime in understanding in biology, essentially how genes are regulated and how they produce something. And among them are things like programs of aging, right? So one of the things that's really quite remarkable about life processes is that every species has its own median age of life and death. And that means there are programs in every one of those species for our lifespan. And we are beginning to understand those programs and how they're coming about and why they're limiting and what happens. And this relates, like everything else, it all relates back to evolution. Evolution is our unified field theory for biology. There is in biology a unified field theory, and we've known it since 1859, since Darwin wrote his book. Right? And that, in fact, if organisms didn't die, they would use up all the resources, and there would be no organisms that live, right? So, and that just shows what Darwin really understood, which is the need for diversity, right? The need to change every generation to respond to the changes in environment and so forth, to keep life optimal for reproduction, right? So let me begin. That's roughly the outline of the course. That's roughly the strategy that we've put together. And as I say, it's challenging for faculty and for students in the sense that you are a very diverse group. So one of the things we thought would be occasionally useful to do is we're pushing you pretty hard. We'll usually go from about 9 to about 10:30, take a half-hour break, go from about 11 to about 12:30, 1 o'clock, 12:30, 1 o'clock, then have lunch, and then come back at 2 o'clock and be finished by 3:30, then have tea, right? And then you could have off, except for today. It turns out we thought it would be really good if we went around the room and have everyone introduce themselves, just to find out who we all are. So we'd like you to all come back. Tea is 3:30 to 4. After tea, it's tea and cookies, and the cookies are good, let me tell you. The Institute for a long time has had a chef, and the chef will, the sous chef makes the cookies. So come back at 4, and what we'll do is go around the room and introduce ourselves. But then also we could take some time to just talk, right? Where if you have questions, and that will be good. And we're going to do that again on Thursday and Friday. What we'll do is we'll take the afternoon. Not everybody has to show up if you don't want to show up. If you want to show up, there'll be time for questions and discussions and interactions with various faculty members. Some of the faculty members are going to stay for the continuum of the course or for 1 week or things like that. So we'll do that for the first week, and we'll do that for the second week. This is actually, I don't know how it is in physics, but in biology, this is a very wholesome tradition, actually. It starts off at Cold Spring Harbor, where you start at 8 in the morning and you go to about 10 o'clock at night in the lab, with a lab and a course. And then what you do after 10 o'clock at night is you go and drink beer and dance. And so there is beer here, but I don't know of any dancing anyhow. So, but it'll have the tradition of a high volume Cold Spring Harbor course. You have the weekend free. For those of you who haven't seen New York or who want to see more of New York, the electricity may or may not be on, but you can get to New York. All right. So that's the course. Let me give you a little bit of a background. I'm going to give you a background about things that are universal to life processes. Right? I thought that may be a good place to start because almost everything I'm going to say will be one of the topics we're going to deal with in the subsequent times. So as I said, really biology as a subject matter, a unified subject matter, begins in 1859 with the origin of the species by Charles Darwin. And Darwin recognized some really interesting things. He's really a naturalist, and he first recognizes what a species is. A species is a freely interbreeding population, right? And outside of a species, there's actually chairs here if you'd like. So you can sit up here or sit in there, yeah. So a species is a freely interbreeding population, right? And as a generalization in Darwin's time, species don't breed outside of their own species. We're finding out that the primates, like Neanderthals and humans, spread, and we always thought of them as different species, but obviously the DNA of Neanderthals are in as much as 4% in Caucasians. And so obviously there's some persistence of this DNA, and the vitality of the offspring wasn't really challenged too much. Although we're learning more about some rather peculiar restrictions about who was the mother of all of this lineage and things like that. And Nick, I don't know if you'll talk about that at all. I mean, I know you've worked on it, but...
Not a bit.
No, okay. Yes, okay, yeah. So Nick is a mathematician who has worked on the Neanderthal genome and how you recognize it in the background of a human, for example. What Darwin realized is they're these species, and he immediately saw that a species has diversity, right? And so he appreciated that within a species there was an enormous distribution of diversity. And as a species arises, maybe there isn't so much diversity, but at each generation, diversity now increases, right? Now this is simply because of something you know from physics right away, and that is if you're transferring information, and everything I'm going to talk about today and we're going to talk about today is information transfer. It's really Shannon theory. It's information transfer. If you're talking about information, you have error frequency. And it's this error frequency from generation to generation that's generating the diversity, right? What he also appreciated was, and this was his enormous insight, was that natural selection acts upon this diversity and will favor anyone in the species who can reproduce the most, leave the most offspring in the next generation. Right? Now, reproduction has many components to it, so it's a very complex set of things. And when we talk about evolution, some of it will come out. And many of the topics we are going to talk about, in fact, bear on reproductive capabilities and fitness of reproduction. But fundamentally, that was Darwin's contribution. But Darwin never really asked the question. In fact, it never appears in any of his writings about what it was that carried the information. What it was that the information was the substances in a cell, in an organism, that carries the information that he's talking about, that shows up from generation to generation to generation and has variation, right? And a contemporary of Darwin, who Darwin never met or never knew, was Mendel. So that's already 1865. So that's only a few years later. Indeed, Mendel, when he did his genetics of peas, Mendel sent his paper to Darwin. And we know that because it's in the Darwin library. But we also know that Darwin never opened the paper, right? So in those days, you had to rip the paper open because of the way it was printed. And Darwin never looked at the paper, right? And he's probably got a lot of papers, so I don't blame him. So, but Mendel, of course, did say something, right? That gave some substance to the idea of transfer of information. First of all, he said, here are the peas and they have traits, right? Traits were phenotypes, essentially, right? And he had to pick what traits were. It wasn't obvious how to pick a simple trait, but he picked color, yellow and green peas. He picked tall and small peas. He picked smooth and rough peas. It's how the skin of the pea or the pod looked. And what he found out was they were simply inherited as a trait. And he immediately realized by looking at the data that traits occur in pairs, right? Those are the so-called diploid organisms. Most of the organisms, animals and plants are more than diploid or more than diploid. And he immediately realized that traits had dominance or recessiveness. And a recessive trait could be hidden by a dominant trait when you have a heterozygote, when you have a plus and a minus. However, it would come back in the next generation in 1/4 of all the offspring. So it was simple ratios of 2s, a simple set of rules. And one of his rules, which I think really shows that Mendel probably had this conception before he ever started his experiments, and he just made his conception true, is independent assortment. Each of these things assort independently. So color and smoothness and height are completely independent traits. They don't go together at all. There's no mutual information. There's no correlation between these kinds of things. It's really quite interesting because Mendel had 7 or 8 traits, and there are 7 or 8 chromosomes. And each chromosome would show linkage, of course, of traits, right? And yet he stopped at the 7, which is roughly the number of chromosomes he had in his pea strain, right? So it may well be he saw linkage, but he didn't like it, and he just withheld it. I mean, for all we know, right? There's always been a kind of suspicion about Mendel's numbers because they're so good, right? In any case, it doesn't matter right then, because at the 20th century, Mendel's rediscovered and shown that what he's done is true for a number of organisms. The one that really takes off is flies. And it's the work of Morgan and Sturdivant. Sturdivant's an undergraduate at Columbia. Morgan is the professor. And they show linkage for the first time. They show recombination for the first time. They really have taken that and turned it into substance. And with time, a lot of the Mendelian rules are violated by genetic tricks, which are now really clear what's happening, but which violate these simple rules. But nonetheless, it was the beginning of understanding what a trait was and what information was. By that time, by the 20th century, people had recognized that there were chromosomes in cells. And they recognized that when cells divide, the chromosomes pair, and then they separate. And they separate in 2 parts. And that looked exactly like Mendel's separation of independent traits. There were maybe 7 chromosomes in the peas, and you could see them and so forth. And so chromosomes were, in fact, the substance that everybody would have agreed turn of the century through 1944. Everyone would agree that chromosomes are probably carrying the information in some way, right? Now, chromosomes were made up of a number of chemicals, but the 2 major chemicals were DNA and protein. And since DNA and protein had been well characterized, people bet on protein. And they bet on protein as the genetic information throughout that whole period of time for the following reason. DNA was boring, right? It only had 4 bases, right, first of all. Secondly, even worse, AO is equal T and GO is equal C, right? So the diversity of DNA didn't look like it was going to handle the information problem that people thought should exist. Amino acids had variable lengths. They had 20 amino acids that were possible. The combinatorics were enormous. And so people were betting on protein all the time. And it was 1944 when that changed. And it changed in a neat experiment that I'll just go through very briefly, and then I'll talk a little bit about DNA and the genetic code, right? In this experiment, there's a bacteria called pneumococcus. Pneumo is lung. Coccus is a round circular bacteria. And pneumococcus was interesting because it could take up DNA from foreign sources, right? And there were 2 pneumococci that that would grow in the Petri dish. They were the same species. They were just the variants in the Darwinian variants that you saw. One made a smooth colony, one made a rough colony. Rough means the edges of the colony were serrate. The smooth one, when put in mice, killed the mice. The rough one, when put in mice, never killed the mice. Right? So that was a kind of phenotype, death in mice. And second was it correlated with the phenotype of smooth and rough. Smooth colonies were smooth because they had a polysaccharide around the coccus, around the bacteria. Polysaccharides are polymers of sugars, and they're very slippery. So that when the macrophages, which eat bacteria and protect the mouse, try to eat a smooth colony or a smooth bacteria, they slip off and they can't get the bacteria into the macrophage. But a rough one they can eat, and then they digest and they kill. And that's the reason why the smooth ones kill and the rough ones don't, right? So what they did was they took DNA from the smooth ones and they put it in the rough ones, and then they put it in a mouse, because that was the selection they used. Was it, a rare one could happen, but it would grow up because it would now kill the mouse. And they found the smooth colonies. So they could transfer from smooth to rough, and once they understood that it was the synthesis of a polysaccharide, there was an enzyme that made that, and then there was a gene that made the enzyme, and a gene that encoded the information for the enzyme. And they really proved that DNA was the genetic material. Now, I have to say, people were enormously skeptical of this is Avery, McLeod, and McCarty at Rockefeller University, or Institute, Medical and Research Institute. People were enormously skeptical of it. But there were second and third and fourth experiments done differently. In fact, the Nobel Prize was given to someone else for that discovery on using a bacteriophage, Hershey and Chase, for example. So DNA was the genetic material. There was evidence for that. And that comes to 1953, which really changes biology and changes this course, and even got us an invitation, us as the biologists, an invitation to be at the Institute for Advanced Study. And that is the Watson and Crick in Cambridge go visit. Watson goes visit Rosalind Franklin, takes a look at her X-ray data, right? Immediately sees that DNA is a helix. In fact, it's a double helix. Immediately gets some measurements about the size of the double helix. And they put together a model of how DNA looks, right? And it is quintessentially, it's first of all a kind of, I always think of it as roughly the kind of paper that's like the 1905 and 1917 papers of Einstein. It has like 6 references in it because there's nothing before it except that DNA is the genetic material, right? And it's a quintessentially great experiment where structure dictates function in a really interesting way. And so I'll finish up with that in a second, right? But one has to say, Rosalind Franklin did the experiment and never got really rewarded. Unfortunately, she passed away before the Nobel Prize was actually given out to Watson and Crick. But Watson and Crick did the modeling, and the modeling just blew up in the field. So I'm just going to finish up by telling you a little bit about DNA, which then David Ting will start talking about how we sequence DNA. The reason to know how we sequence DNA is this isn't like a measurement of 7 decimal places. There are probabilities of errors in DNA sequencing that are high. And remember, I've told you all about diversity, which means there should be differences between DNA of 2 different people. And how do you tell the difference between a mistake, and how do you tell the difference between a real difference between people, right? And of course, you use information theory, and you just keep sequencing until you go far enough that you're either blind or you can figure out what's happening, right? All right, so I'm going to pause and just take you back, because the one thing I've learned at the Institute is that physicists are very poor, this is not an insult to anybody, but very poorly trained in chemistry, right? So for some reason, physicists don't take chemistry except quantum chemistry, right? So let me get you to a nucleotide, right? So this is adenosine. This is adenylic acid. It's made up of a base. This is a purine base. Pyrimidine base has only 1 ring with 2 nitrogens in it. And then this is deoxyribose. It's deoxy because this is a hydrogen here, right? And the numbering system is what I want to just get you familiar with. This is the thing I think that not many of you would recognize. You start numbering the bases, the carbons of this sugar. There are 5 of them. There are 5 carbons. One is here, 2 is here. So deoxy is 2. If that's a hydroxyl group, that's ribose, right? And 3 is here. That makes it deoxyribose. 4 is here. 5 is here, right? And so the base comes to 1, and the phosphate groups comes off 5, all right? Now, this is one subunit of a polymer, and the polymer is a 3,5 phosphate polymer. So the next phosphate group will come off here, right? And so there's a polarity to DNA, a 3 to 5 polarity in one direction and a 5 to 3 polarity in the other direction of what's at the ends, right? There's either 5' triphosphate at the end or 3' phosphate at the beginning, right? There's a real polarity. There's one other thing to say. It's very one of the things we left out of this course, maybe we could talk about it some afternoon if you want, is the origin of life question. It's a very interesting question. It's just very hard to prove. No one has actually recreated any life form in the lab. And and even if you did recreate a life form, in fact, Clyde's lecture this afternoon will be called "What is Life?" And it's a very hard thing to prove that you have created life the same way that the origins of life came about. So it's mostly speculative. It's a really interesting question, but we haven't added it for that reason. There are other things we haven't added that are good. But most people are going to say, as you'll see when we look at the genetic code, right, that the information in organisms at the beginning was held by RNA, right? And this information is held by RNA, right? Because not only is it a way to hold information by the sequence of nucleotides, right? But it is catalytic. So 2 hydroxyl groups here are very reactive. And you can make a catalyst out of the information, right? Now, on one hand, that is 2 jobs for the price of 1. On the other hand, you're destroying the information when you act as a catalyst, right? And that isn't tolerable if you want to have stable information that passes along. So that looks like RNA is the start, and then it changes over to DNA because it's more stable. All right. So let me just take you through this. This is what you already know. This is the 5 to 3' polarity and the double helix of DNA. There's large grooves and small grooves in the DNA where proteins approach and recognize the base pairs, right? And this is what Watson and Crick published, right? And they made 2 hypotheses that were really neat, right? The first hypothesis that they made was that the 2 strands would separate, right? And that's how replication occurs. Because A pairs with T and C pairs with G, right? Purine and pyrimidine. Yes? Yes. Remember I said the 5' phosphate?
Yeah, I saw it. I was looking at it. I couldn't see full carbon. I feel like I missed a really crucial thing.
Oh. No, you did only see 4 carbons because chemists don't put carbons in, right? So that's 1, right? That's 2. That's 3. That's 4. That's 5. Actually, what this looks like without the phosphate is a hydroxyl group, right? And it's carbon hydroxyl group. And then when a bond is made, it's a phosphodiester bond, right? All right, so the first prediction they make is the strands will separate, right? That's 1953. By 1957, the prediction is tested. So the way it's tested, I'm going to go fast because I'm already going to pull on your time. The way it's tested is as follows. It is possible to centrifuge DNA or proteins in a density gradient, right? So what you do is you'd use a salt like cesium chloride. And cesium chloride gradients are made by putting cesium chloride in a tube and then spinning it, and then the heavier cesium goes to the bottom, and you get a density gradient, right? And then the DNA bands at a particular density, mass per unit volume, right? Okay. So if I grow bacteria in not C12, but C13 carbon, right? That's the glucose would be C13 carbon, and N15, not N14, right? I'm adding heavy atoms to the glucose and the nitrogens that ultimately go in DNA, right? And then if I put that in a density gradient, it will have a different density because it's heavier, right? It's mass per unit volume. Unit volume stays the same, but its mass per unit volume it has changed, right? So if I take just bacteria grown in normal water, normal carbon, normal nitrogen, it has what I'll call, because there are two strands, light density, right? Light density. If one generation goes by in heavy medium, it will be light heavy, right? And that will show that the strands have separated and one will be heavy and one will be light. By the next generation, I should see for the first time heavy, right? Okay, so that sounds great. And they did that. This is an experiment done by Messelson and Stahl at Caltech, right? And they did that and they got that answer, right? In fact, they got an answer which was more than they ever expected, right? They actually saw heavy before they finished the heavy light. Now, I could leave that to your imagination as to how to interpret that, right? But it really just to think about it, if you see heavy before you ever finish a chromosome, it means you're starting at a particular origin and you're starting a second replication round before you finish the first, right? Do you follow? So it's not that, and there's a reason for that, and it'll be on the next slide, in fact, okay? Next or the next last slide. All right, so that's the first prediction. And the second prediction is that the sequence of nucleotides in one of the strands will predict what amino acid there is in a protein, right? Information transfer, right? And there, by 1961, the first codon, that is the first sequence that tracked an amino acid, was known. They made a polymer of U. U, by the way, is the ribose form of it, of T, right? So they made a polymer of U's, right? They then looked at the amino acid, and it was phenylalanine. They made a polymer of A. They looked at the amino acid, and it was lysine, right? And so they just translated everything in vitro on ribosomes. And that's the work of that's the work of Nuremberg and Mattei. And these are two, well, Nuremberg and Mattei anyhow got Nobel Prize for that. And that broke the genetic code. They worked out all 64 codons. The genetic code was a triplet. Had triplet of information for every amino acid. Okay, so let me just give you some scale here. So now you know that the predictions made by Watson and Crick came true. They came true very fast, by 53 to 61, 53 to 57. And molecular biology as a substance and as a subject matter started, right? Okay. Now, measuring DNA contents of organisms immediately gave us several paradoxes, which we'll talk about throughout the week about how they handled, right? So E. coli is a bacteria. It's about a micron long. It's a rectangle, and it's about a micron long, right? And its size, 1 to 2 microns. E. coli DNA has 4.6 times 10 to the 6 base pairs. So it's 1 million base pairs. That's the amount of information that it has. And that translated, now that it's been completely sequenced, to about 3,000 genes. The units of function, that's the units that make amino acids. There are genes also that make RNAs, right? The size of this DNA is about 1.6 millimeter. And that brings up the first problem. How do you stick 1.6 millimeters in something that's a rectangle that has about 4 times 10 to the 6th DNA, 1.6 millimeter? So there's a packaging problem, right? Packaging problem gets more extensive with human DNA. It's 8 to 12 micron. That's a human cell. It's about 8 to 12 microns. 3 times 10 to the 9th base pairs, 25,000 genes. So you already now know that there's a nonlinear function between DNA and genes, right? And how does that happen? And what does it mean? That's something that Ben and others will address today. It means there's other DNAs there that are not genetically coding for substances called your genes, but are there. And they may have functions and they may not, and that's a problem we keep addressing. So there's a nonlinear relationship between DNA and gene numbers. This is even more extreme. It's about 2 meters of DNA. And so we got big packaging problems. And especially if you want to take that DNA and open it up to replicate, or you want to take that DNA and open it up to read it, right? Transcribe it. And the packaging problem is solved by something we're not addressing in the course again, chromatin structure, right? But we can talk about it if anybody wants to bring it up, or right? This extra DNA is often repetitive DNA, but it also is the fact that there's nonlinear information in the chromosomes. That is to say, there's information to make a protein, then there's information that doesn't make the protein, then there's information to make the same protein, and that brings up a whole new process called splicing of the messenger RNAs. Again, something we're not going to talk about in great detail. And finally, let me get to something which came from the genetic code, right? This is the triplet code. First base, UCAA, at second base and third base, right? So there's a few things to notice, and these are the 20 amino acids. The first thing to note about it, well, if you have 3 bases, right? And you have 4 bases and you have 3 spots to put the 4 bases, you have 64 codons. And so these are the 64 codons. And the first thing to say about the 64 codons is that they are degenerate, right? That is to say, there are some codons, leucine is a good example, that there are 6 codons for leucine, right? There are 6 codons for arginine, right? There are 2 codons for serine. There are 4 codons, oh sorry, there are 6 codons for serine, right? There's 4 here and then there's 2 here, right? But proline, threonine, and alanine have 4 codons. There's some that have 3 codons. There's some that have 2 codons. And only methionine and tryptophan have 1 codon. Is that right? Is it tryptophan? Yes, only tryptophan and methionine have 1 codon, right? So the codons are degenerate. Most interesting, they're almost 90% of the degeneracy is in the 3rd position. So codons have positions, of course, because they're read in an order, right? So the 90% of the degeneracy is in the 3rd codon. This has led to the idea that maybe the first code was a 2-letter code. And it only could encode 16 amino acids. And so it expanded to encode 20, or that was the optimal number, presumably. Now we're speculating, of course, right? And so the first genetic information might be RNA, and the first genetic code might be 2 codons code, right? And degeneracy is a product of that. But it's not ambiguous. That is to say, there is no codon that encodes 2 different amino acids, right? So it's not ambiguous. And most interesting of all, it's universal. There has been no organism sequenced on Earth that didn't use this code, right? And that's a very interesting observation. Unfortunately, it isn't. You could jump to the simple conclusion that there's one origin, right? And the origin fixed the code, and then it moves on, right? But that's probably not the only conclusion you should jump to, because it's possible there were multiple origins, and they ended up competing against each other with multiple codes, and this is the best of all possible codes, right? Now, if it is, we can't tell you why. Or I can't tell you why. Let me put it that way. Maybe you guys could figure out why. Why this would be the best of all possible codes. There is a kind of buffering in this code. It's a chemical buffering. For example, these are the hydrophobic amino acids on this side of, right? So one amino acid change has a tendency to change hydrophobic to hydrophobic. And that would minimize the functional change in the protein, presumably. These are the charged amino acids, right, are in here frequently. And so basic and acidic amino acids tend to be a bit exchangeable with one amino acid change. So there's a kind of buffering, but that's the closest anybody can sort of pick. There are 3 stop codons. A stop codon means end of protein. There is no amino acid equivalent that is put in, and it stops. And there's 1 start codon, that's methionine. Every protein usually starts with methionine. You may not see that when you look at the protein, but that's because a protease has clipped the methionine off, right? But nonetheless, it is always made with a methionine. So it's a start codon. So there is a start, a stop, there are 3 stops. And the code is universal is really the most amazing thing. What's really neat is that presumably when we get to Mars and we find an organism and we sequence that organism, we're going to find it has the same genetic code, right? And then there are 2 possibilities. One is it was contaminated by an earlier trip to Mars by, and this is just a, yeah.
Life could have originated on
Exactly. That's right. That's right. Maybe. Well, so there is a theory which Crick liked a lot called panspermia, which is a theory that life starts way the heck out in the universe, or well outside in the universe, and comes and spreads on things like comets and things like that. So, but the universality of the code in here is a really interesting final conclusion. So I wanted to give, that's the sort of wrap up of things about biology that are universal, right? From here on in, it's all downhill. So there's no universality of anything. So I took the easy lecture. But we're actually going to now here sort of get in our teeth into how DNA sequencing is done as a fundamental background for everything that's going to follow as we start looking at information transfers. Okay, so this is David Ting. He's a professor in oncology at Massachusetts General Hospital, and he does many different things at Massachusetts General Hospital, but one of them is he runs the sequencing facility. Right? So David.